Hello everyone. In this lecture we are going to discuss about carbohydrate classification. So in the last lecture we have seen about introduction to carbohydrates and outline classification, and also we have seen about monosaccharides classification and about trioses. So this is the lecture 2 of carbohydrates. So before going to in detail classification, we need to discuss some basic definitions and important definitions. So we have to know what are asymmetric carbons here. So asymmetric carbon atoms are those atoms which contains 4 different atoms or group of atoms are attached. So if you can see the structure of a glycerose, so this is the central carbon atom in the glycerose structure, and if you observe the groups around the carbon atom in the glycerose, you can see the 4 different atoms arranged. So at the top you can see the aldehyde group, at the bottom you can see the CH2OH, and either side you can see the hydrogen and hydroxyl group which are different to each other, and these are the 4 different groups which are actually arranged around the carbon atom in case of glycerose. So in glycerose we can say this carbon atom is an asymmetric carbon. Okay, so here, and the compounds which contains this asymmetric carbon are known as asymmetric compound. So we can say that glycerose here is an asymmetric compound as it consists of an asymmetric carbon in its structure. When you take the example of dihydroxyacetone, when you see the structure here, so it consists of CH2OH at its top and bottom. The structure, it consists of a ketone group which doesn't resemble 4 different groups. So it don't have any asymmetric carbon like glycerose. So as I have mentioned, the compound containing an asymmetric carbon atom is called an asymmetric compound. The example will be the glycerose. So the main property of the asymmetric carbon compounds will be the formation of isomers. So the presence of asymmetric carbon atoms in a carbohydrate produce the isomers. So what are isomers here? Isomers are the different compounds with the same molecular formula but difference in their arrangement. So if you see here, glycerose. So glycerose consists of one asymmetric carbon atom, and so due to the presence of this asymmetric carbon atom, it can form the isomers. So here the number of isomers which a compound forms is also depends upon the number of asymmetric carbon atoms. As you can see here, if the compound contains one asymmetric carbon, it will form the 2 isomers. If it contains 2 asymmetric carbon atoms, it forms the 4 isomers. So here the very important point which you must note here is the number of isomers depends on the number of asymmetric carbon atoms present in a molecule. So you can remember with this formula 2 to the power n, the total number of isomers of a compound will be equal to 2 to the power n, where the n represents the number of asymmetric carbon atoms. Say for example, if you take the example of a glycerose, when you observe the structure of the glycerose, so the glycerose consists of only one carbon atom in around which 4 different atoms are attached. So if you substitute the number of asymmetric carbon at the one where n is equal to 1, N is equal to 1, which is the one asymmetric carbon. When you substitute here 2 to the power 1, 2 ones are 2. So hence the glycerols can form two different kinds of isomers which are mirror images. So in case of D-glycerols, the arrangement of hydroxyl and hydrogen group will be like this. In case of L-glycerols, it will be like yes, vice versa. So it form mirror images which are the two isomers in case of glycerols. I hope you understood about asymmetric carbon atom and about the isomer formation. So simply you can remember the presence of asymmetric carbon is equal to number of isomer formation. So if the molecule don't have any asymmetric carbon atom, so no optical isomers will be formed. So if you can see here, if you observe the structure of a dihydroxyacetone, so it don't have no asymmetric carbon atoms are present here, is it? So that's why there will be no isomer formation in case of dihydroxyacetone. So if the molecule don't have any asymmetric carbon, hence you can prove that it won't be forming any isomers. So these are some basic points about asymmetric carbon and what are isomers, how the isomers actually form. So tetroses, which comes under the monosaccharides. So the tetroses consists of 4 carbon atoms in their structure and the molecular formula will be C4H8 and O4. If the tetroses contains an aldehyde group in its structure known as aldotetrose, if it contains the keto group known as ketotetrose or tetrulose. So when you observe the aldotetrose, the most common form of aldotetrose occurring in organism is erythrose and the common tetrulose is the erythrulose. So this erythrulose appears in the form of its phosphate as an intermediate in several biochemical transformation. So if you observe some metabolic pathways in plants like Calvin cycle, you will tend to see this kind of erythrulose compounds in its pathway as intermediate products. So here erythrulose is a ketotetrose which you can see in some metabolic pathways or biochemical transformation in case of plant bodies. So if you observe the structure here, so this is the structure of an erythrose. If you observe the structure of an erythrose, it consists of two asymmetric carbons at position 2 and 3. When you observe here, it consists of 4 different atoms arranged around itself. So which forms an two asymmetric carbon atoms at position 2 and 3. So due to the presence of two asymmetric carbon atoms, when you substitute here 2 to the power n is equal to 2 here, which means two asymmetric carbons are present. So 4 isomers are possible in case of erythrose, which is D-erythrose, L-erythrose, D-threose and L-threose. So you can see the difference in alignment. So the arrangement is like mirror images between D and L erythrose and D and L threose. So this is about tetroses. So when you observe the structure of an erythrulose, it consists of only one asymmetric carbon atom at position 3. So where you can observe like this. So due to the presence of only one asymmetric carbon here at carbon atom T3, you can observe it forms two isomers which are mirror images to each other. Okay, so different functional groups are arranged like this: C double bond O, CH2OH, H and OH. So these are the four atoms or groups arranged around the carbon atom of an erythrulose. So due to the presence of these four different groups, differentiates this carbon atom as asymmetric carbon atom in erythrulose, leads to the formation of two different kinds of isomers. Next, pentoses. The pentoses consists of 5 carbon atoms in its structure, and the molecular formula will be C5H10O5. If the pentose contains aldehyde group, it is known as aldopentose. If it contains keto group, it is known as ketopentose or pentulose. So there are some biologically important pentoses are present: ribose and deoxyribose. So ribose sugars can be observed in RNA, the composition of RNA, ribonucleic acids, and DNA, deoxyribonucleic acid, contains the composition of deoxyribose. So these are the well-known pentoses, which are ribose and deoxyribose. So if it in case of xylulose, xylose and ribulose, so again you can see this xylulose, xylose and ribulose in case of plant metabolic pathway in Kelvin cycle. So these are some most biologically important pentoses. So ribose phosphates occurs in some coenzymes like L-xylulose is a metabolite of glucuronic acid and is excreted in urine of humans affected with hereditary abnormality in metabolism called pentosuria. So among this, the ribose and deoxyribose, xylulose, you can observe these pentose sugars in humans as well. So ribulose you can found in the plant metabolic pathway like Kelvin cycle. So if you take the example of L-xylulose, it is a metabolite of glucuronic acid. So it comes as a byproduct in the metabolism of glucuronic acid, which will be excreted in the urine of humans affected with an hereditary abnormality in metabolism called pentosuria. So